Hello students, in the last video we started the chapter 2 of the class 12 biology. In that we have seen pre-fertilization events. The pre-fertilization events are divided into 4 steps, that is male reproductive structures and event, second female reproductive structures and event, third one pollination, and fourth one is pollen-pistil interaction. In male reproductive structures and event, we have seen the microsporogenesis. What happened in the microsporogenesis? The microspore mother cell will converted into microspores. That is called through the meiosis. That is called microsporogenesis. Here, microspore mother cell that will by meiosis it will convert into microspore tetrad. This microspore tetrad, during the maturation, it will get separated and 4 pollen grain will get separate. So this 4 pollen grain from here to the pollen grain formation, or we can say the microspore formation, is called microsporogenesis. This microsporogenesis will follow by male gametophyte development. During the male gametophyte development, from each microspore or pollen grain, the formation of gametes will take place. So here I have shown one pollen grain. As I said earlier, pollen grain has got 2 walls, inner wall and outer wall, intine and exine. Exine is made up of sporopollineum. This sporopollineum gap is there, we can see over here. It is called the germ pore. If it has one spore, it is called monoclopate. If it has got 2 pores, it is biclopate, and if it's got 3 pores, then it is called triclopate. Here you can say there are 3 pores. The germ pores I am talking about. 3 germ pores, hence it is called the triclopate pollen grain. This triclopate pollen grain then followed by the meiosis. What happened in the mitosis? What happened in the mitosis? The ploidy of the cell will not get changed. Here, during the microsporogenesis, diploid cell has converted into haploid form. Now the pollen grain is haploid. Again the mitosis will take place, which will have the same haploid cell. So here, as you see, 2 cells are formed, and out of those 2 cells, the smaller cell that is known as the germ cell, and the nucleus present into the germ cell is called the germ nuclei. The larger cell that is called the vegetative cell, and the nucleus is called the vegetative nucleus. The vegetative cell is also termed as tube cell, and the nucleus is tube nucleus. Now what happened at this stage? Almost 60% of the angiospermic flowers that will follow a mitosis. And during that mitosis, this germ cell nuclei will get converted into 2 nuclei, and these nuclei are called the gametes. But in rest of the plants, around 40% of the plant, the pollen will get released at 2 nuclei stage, and once the pollen get laid down on the stigma, they will form pollen tube. So here, the vegetative cell or tube cell will form a pollen tube, and as it forms the pollen tube, this pollen nuclei will be there in front, and the germ cells will be behind them. In 60% of the plant, this stage you can see, and in rest of the plant, this nuclei, can germ nuclei will get divided into the pollen tube. The female reproductive structure is called megasporophilous, carpel or pistil. Carpel or pistil we can say, or we can say the megasporophilous. This carpel is made up of 3 different parts: stigma, style and ovary. In some plants, the carpel is only one, hence those are called monocarpous. If more than one carpels are found in any flower, that is called the polycarpous. These are the structure of monocarpous, apocarpous and syncarpous. The single carpel is called monocarpous. Polycarpous, but ovaries are not attached, the carpels are not fused, that is called apocarpous. Here the example of apocarpous is mycelia. Then in syncarpous is a kind of polycarpous, and where the ovaries or the carpels are fused, that is called syncarpous. The example is papaya, or we can say the poppy flower. Now we understand the structure of ovule. In ovule will start with the funicle. This is the part which attach inside the ovary. And the part from where the integument starts developing is called the chalaza or chalazial end. Then the end, the integument's end, where it makes one pore, it is called the micropylar end. And inside this integuments, it is filled with nucellus tissues. This whole part is called the nucellus, and in that nucellus there are plenty of cells are present. Out of these cells, one cell from the micropylar end will get differentiated into megaspore mother cell. Here you can see this cell. Suppose from here, if any cell will convert or will get differentiated into megaspore. This one is the megaspore mother cell. Development of megaspore from megaspore mother cell is called megasporogenesis. Microsporogenesis takes place in male parts, and the megasporogenesis is a part of the female reproductive system. Megasporogenesis starts with the megaspore mother cell. This megaspore mother cell is initially uninucleated and will convert into 4-cell stage. This, you can see, this one out of these 4 cells, one is large in size and which is active megaspore. So megaspore mother cell will form 4 megaspore tetrad. We can say the megaspore tetrad. This, out of these 4 cells, 3 cells will get degenerate. Only 1 cell will be there to form the gametophyte. This megaspore we have shown over here. So from here megaspore mother cell to megaspore formation, mature megaspore formation, it is called the megasporogenesis. Megaspore will follow the mitosis and will form 2n number of haploid nucleus. If you see this one, if it's present in here, so this is the chalazial end and this is the micropylar end. So here this will go to the chalazial end, this will go into the micropylar end. Now again mitosis will take place for the 2 times and it will form 8 nucleated stage. In this 8 nucleated, we can see there is not proper organization, and after this 8 nucleated stage, what will happen? This nucleus will be covered with the cell wall. So the cell wall formation will take place. This after cell wall formation, they will form themselves in a proper arrangement. During the arrangement, out of this 8 cells, 3 cells will get migrate, will get settled at the chelazal side. Those are called the antipodal cells. Whereas 3 cells will get migrate and settle at the micropylon end. This is out of these 3 cells, 2 cells will form the synergids and 1 is a egg cell, and the remaining 2 cells which will be settled in the middle, those 2 cells will get fused with each other and will form a single cell which have 2 nucleated. We can say the polar nuclei, or you can say the central nuclei. Hence, this stage is having 8 nucleus and 7 cell stage. This is the chelazal end and this is the micropylon end. 3 cells will rearrange and get settled over here and form antipodal cells.